Introduction. In the field of biochemistry and molecular biology, understanding the intricate processes involved in translation is crucial for advancing our knowledge of cellular functions. Translation is the fundamental process by which ribosomes decode messenger RNA, mRNA, molecules and synthesize proteins. However, there are various complex aspects within translation that contribute to its overall efficiency and accuracy. This script aims to delve into 12 essential topics related to the world of translation. Each topic will provide detailed insights into key mechanisms, molecular events, and regulatory processes that significantly impact protein synthesis. Through an exploration of tRNA modifications, ribozymes and catalytic RNA, alternative splicing, protein targeting, non-canonical amino acids, ribosome structure and dynamics, translational control in diseases, frame shifting, mRNA localization in neurons, translational fidelity, quantitative analysis of translation rates, translational regulation by microRNAs, we will gain a comprehensive understanding of this vital biological process. Now, let's embark on this journey of discovery and unravel the fascinating complexities behind translation. The process of transferring genetic information stored in mRNA to protein is largely dependent on tRNA molecules. tRNA plays a crucial role in decoding mRNA sequences and delivering the correct amino acids during protein synthesis. However, tRNA undergoes multiple modifications to ensure its proper functionality. These modifications include the addition of methyl groups, pseudouracilation, and base modifications, among others. These alterations, catalyzed by enzymes, are required for accurate decoding and efficient translation. The diverse range of modifications provides additional structural stability to tRNA and contributes to its recognition by various translation factors and ribosomal components. The concept that RNA can possess catalytic activity challenged the long-held belief that only proteins could act as biological catalysts. Ribozymes, RNA molecules capable of catalyzing specific chemical reactions, have been identified in various biological processes, including translation. These ribozymes, such as the self-splicing group I intron, the hammerhead ribozyme, and the ribonuclease P, can perform essential functions during translation initiation, elongation, and termination. Understanding the three-dimensional structures and mechanisms of ribozymes provides critical insights into the evolution of early life on Earth and the potential applications in biotechnology and therapeutics. Alternative splicing is a complex post-transcriptional process that generates multiple mRNA isoforms by selectively splicing exons of a pre-mRNA. This phenomenon significantly expands the diversity of proteins encoded by a limited number of genes. Alternative splicing also affects translation by introducing different open reading frames, altering the coding sequence, or introducing premature stop codons. Furthermore, regulatory elements, such as exonic splicing enhancers or silencers, influence mRNA splicing decisions, which in turn determine the abundance and functional characteristics of the translated proteins. Proper protein targeting and localization ensure that newly synthesized polypeptides reach their intended subcellular compartments. Targeting signals, such as signal peptides and sorting motifs, direct proteins to the endoplasmic reticulum, mitochondria, nucleus, peroxisomes, or other organelles. The process of co-translational or post-translational targeting involves the involvement of signal recognition particles, translocation channels, chaperones, and receptor proteins that facilitate the transport and localization of proteins. Dysregulation of protein targeting and localization can lead to pathological conditions and cellular dysfunction. Although proteins are typically composed of the 20 canonical amino acids, nature also incorporates non-canonical amino acids into polypeptides, expanding the chemical diversity of the proteome. These non-canonical amino acids are often introduced through distinct tRNA aminoacylation mechanisms or post-translational modifications. The incorporation of non-canonical amino acids allows for site-specific protein labeling, broadens the range of protein functions, and enables the synthesis of proteins with enhanced properties. Understanding the incorporation and utilization of non-canonical amino acids opens up new avenues for protein engineering and therapeutic interventions. Ribosomes are large complexes comprised of ribosomal RNA, RNA, and proteins, responsible for catalyzing peptide bond formation during protein synthesis. The high-resolution structures of ribosomes obtained through cryo-electron microscopy and X-ray crystallography techniques have revealed intricate details regarding the arrangement and interactions of the ribosomal components. Furthermore, advances in single-molecule techniques have provided insights into the dynamic conformational changes occurring during translation, including ribosomal translocation, ribosome recycling, and the proofreading mechanisms that ensure accuracy and fidelity in protein synthesis. Dysregulation of translation plays a significant role in many diseases, including cancer. Abnormal activation or inactivation of translation factors, regulatory RNA molecules, or components of the translation machinery contribute to uncontrolled cell proliferation, tumor angiogenesis, and metastasis. Targeting the translational control mechanisms associated with diseases holds promise for the development of novel therapeutic strategies and the identification of therapeutic targets. Understanding the intricacies of translational control provides insights into how cells respond to environmental cues and regulate protein synthesis to maintain homeostasis. Frame shifting is a regulatory process occurring during translation when ribosomes shift to a different reading frame, leading to the synthesis of distinct protein products. Programmed ribosomal frame shifting often involves specific RNA structures, such as pseudoknots or slippery sequences, that induce ribosomes to change the reading frame. This mechanism allows for the synthesis of multiple proteins from a single mRNA. Frame shifting plays critical roles in viral protein synthesis, genetic recoding, and the production of regulatory proteins involved in cellular processes and disease. Neuronal cells exhibit unique spatial arrangements of gene expression, including local translation of mRNAs within dendrites and axons. This localized protein synthesis allows neurons to rapidly respond to extracellular cues and drive synaptic plasticity. Specialized RNA-binding proteins and regulatory elements ensure the spatial and temporal control of mRNA localization, translation, and degradation. Dysregulation of translation in neurons is linked to neurological disorders, such as Fragile X syndrome and Alzheimer's disease. Unraveling the mechanisms underlying translation and mRNA localization in neurons holds significant implications for understanding brain development and function. Translation fidelity refers to the accuracy of deciphering mRNA instructions to ensure the synthesis of functional proteins. However, errors can occur during elongation, leading to the incorporation of incorrect amino acids or premature termination. These errors, known as mistranslation or translational errors, can be caused by mischarging of tRNAs, inaccurate codon recognition, or ribosomal stalling. Mistranslation has been implicated in various diseases, aging, and cellular stress responses. Investigating the factors and consequences of mistranslation contributes to our understanding of cellular physiology and disease pathogenesis. Determining translation rates is crucial for deciphering the regulation of protein synthesis and understanding the dynamics of cellular processes. Advances in high-throughput techniques, such as ribosome profiling and mass spectrometry, enable the global analysis of translation kinetics and the measurement of protein synthesis rates. These quantitative data provide valuable insights into translational efficiency, the impact of cis-regulatory elements on translation, and the response of translation to cellular conditions and environmental stimuli. MicroRNAs, miRNAs, are small non-coding RNA molecules that play essential roles in post-transcriptional gene regulation. They bind to target mRNA molecules and modulate translation by inhibiting protein synthesis or promoting mRNA degradation. miRNAs contribute to diverse biological processes, including cell proliferation, differentiation, immune responses, and development. Dysregulation of miRNA-mediated translational control has been implicated in various diseases, making microRNAs a promising therapeutic target. Understanding the specific mechanisms and regulatory networks involving miRNAs provides valuable insights into gene expression regulation and potential therapeutic interventions. The process of translation reveals a multifaceted interplay between various molecular components, regulatory mechanisms, and environmental cues. It is evident that translation is not merely a linear process but a complex network of interactions and controls that dictate the fate of protein synthesis. By exploring the 12 topics discussed in this script, we have gained in-depth knowledge and appreciation of the sophistication underlying translation. As future medical professionals, understanding translation and its regulation in health and disease empowers us to contribute to advancements in diagnostics, therapeutics, and disease management. The continuous unraveling of the intricacies of translation will undoubtedly uncover even further mysteries and pave the way for innovative research and therapeutic strategies in the realm of biochemistry and molecular biology. So let us embark on this exciting journey of unraveling the mysteries of translation and its pivotal role in cellular life.